how the blood flow slightly changes and something new is added to their experience, then you can presume that the bit of brain in which the blood flow increases is the bit of the brain which was most turned on by whatever the new thing is that you're going to do. So, if you show someone in the scanner, look at a television screen or in the scanner, just show them a static array of dots or lines or something, which is static. And you compare that state of the brain with not the seeing pattern, just lying in the scanner, then you see, you're familiar with these sorts of pictures, they're everywhere nowadays. It's a horizontal section of the brain. Here's the front of the brain, the back of the brain. And here, this red stuff is where there's been a very small but detectable increase in blood flow. And it is this classical visual area which the 19th century neurologists knew about. So that's fine. Look aesthetically. But if you make them move around the dots and you compare what happens to the brain when you do this, compared with subtract what happens when you do this, you can see any areas that are specifically turned on by movement rather than static things, and it's a very clear result. Other regions further forward, just over the ears here somewhere, called the fifth visual area or the middle temporal area light up. And this disappears because you've subtracted it out. Because you subtract from I mean, this does stimulate that, but no more than static patterns do everything. This is an area, I mean, it's presumed, and the evidence is overwhelming, corresponds to a region which has been well described in monkeys. The neurons, individual neurons in that area respond when the monkey sees a moving object, and each cell responds to a particular direction of movement. You can inject a local anesthetic into a conscious monkey into that region and interfere with its ability to detect movement, and beautiful correlation between what goes on in that area and the monkey's perception of the world, behaviour towards the world. Very nice example. MT. Equally, there's papers every week describing different sorts of functions that are known now to be thought to be localised in the human brain. So, for instance, if you put people in the scanner, and this is the beginning of an experiment done by Tim Andrews in my lab, although part of it described as not novel, he built on that to look at other things. But you put people in a scanner and show them a series of photographs of objects or faces, randomly presented, real faces or man-made objects, one after flashing out for 10 seconds each. And then just said to the computer, tell me parts of the brain which are, on average are more turned on by these things and vice versa. And this time is illustrations. Localization. Here's a section of the brain, a little bit lower down now. This part is actually at the top of the temporal lobe. These are the eyes. And this is a region which was selectively more activated by face patterns than by objects, more stimulated by objects. The faces quite close to each other, 2 or 3 centimetres away from each other. Small little patches. Right-hand side of the brain, not the left side of the brain. And he went on to show, very nice experiment, show that the activity of this region, the variation of activity of this region, could predict with pretty high reliability, like 90%, whether the person was actually consciously perceiving a face or not, when looking at images that were ambiguous. So the association cortex is now thought to be absolutely full of these discrete little areas, all reanalyzing information from the senses or preparing different aspects of motor control. And one third of the cerebral cortex in humans is devoted exclusively to visual analysis. Even though all the information comes in at the back from the eyes, it then gets redistributed amongst all these different areas and reanalyzed again and again. The important point here is that, and it is important, if you start with input to a region and they just shunt it around the brain, there cannot be any gain in information. Information is absolutely limited by whatever gets in the first place. You can't create new information. So you might say, well, I have these extra things. It turns out that although the information is present, it might be present only, it might be invisible to the neurons within the visual area because it depends on knowing what's happening amongst, between the spatial and temporal patterns of information in the primary visual area. So, for instance, recognizing a face involves detecting shapes and forms and gradations of brightness and so on in many different parts of the visual field. So neurons in different regions of this area, which is mapped according to the visual start to see how the brain might work as a spatial computing machine and what these additional areas might be involved in. Here's a sequence of areas. This is the result of a series of scanning experiments in which different sorts of things were seen by or done by, seen by, in this case, subjects, showing these different regions, visual regions, right? It's a sequence to the brain. We know that the static analysis of visual patterns is performed by the fact that's in colour, and this wasn't actually described in this experiment. Further forward, there's several regions. This is certainly V5 or MT, the region I've just described. But there's another strip, V3 principally, involved in the preliminary analysis of movement, so response to moving things like that. And MT can, or the neurons there, can do really quite complicated things like recognising separate patterns of movement between transparent and superimposed moving surfaces. So all of that kind of relatively high-level motion analysis is performed there. The green area in front is an area which is turned on specifically when people look at a very particular sort of moving pattern called biological motion, a pattern like that. It's computer generated, you see it instantly as if it is a walking figure. And all that you're looking at is the motion, relative motion of the articulation, of a few articulation points in the body, and you immediately see the whole form. And there's lots of evidence that is being done by special processes which are distinctly separate from the analysis of simple translation. So we have mechanisms in our head specifically for looking for the kind of motion produced by an articulating organism. Obviously a biological value, behavioural value. Okay, so here's the challenge. What do you think this is? This area here, the purple area, which over the yellow is the overlap to right in front of the biological body, you can see this sort of sequence, static things, ordinary linear movement, complex movement, biological motion. Any guesses?
Danger or fatal or
People we know. People we know is sort of down here. Is it
friend or foe?
Facial expression. Very good speculation used in the experiment. Listening to verbs or reading verbs. And not just verbs of motion. First, it was talking verbs of motion first. But the majority of verbs have got something to do with these surrounds. So it looks as though the analytical machinery for that aspect of language would be tagged on to a processing stream concerned with seeing. And equally, in the parts of the visual system down here which are concerned with objects, you find regions which are activated by adjectives and by nouns, as if you were holding a Venniger's area. This is part of Venniger's area, you see. All the Venniger's area is as it were parasitic on, attached to visual processing. So, from an evolutionary perspective, you can see how that might make sense. That those have all been processed to a point, having a little bit more nudging and a bit more space between the neurons to convert them into analytical machinery aspects of language. Right, let's see then. Let's ask about evolution. This is the assumed tree of heritage of human brains from the common ancestor of the marsupial central mammals. And the areas that these human brains are down, they're not drawn to scale. So this is a hedgehog brain, which is 9 to this, the human brain top here. But the colouring indicates similar function. The S areas, the red and the yellow, are somatic, touch sensory areas. So there's the human somatic sensory strip that I mentioned to you. Auditory areas green, there's the green humans. Visual areas are blue or black. Right? That's empty. And what you see is that if you look at the hedgehog brain, which is probably as close as we can get to the common origin of mammals, it's a tiny brain, and it's almost completely full of sensory areas. There's a motor area in front of it. But it's almost all full of primary sensory areas. And they're arranged in generally the same sort of pattern as in the human brain. Visual here, back, auditory, somatic sensory. They all are. They've all got that same basic organization. And what happens during evolution with this evolution is that the rest of the space, the association areas, get bigger. And we know that in humans the association, and here's cats, the association areas are full of all these extra processing modules. So evolution seems to have added new computational modules. That's kind of added a new PC and another one, and a microprocessor, just to cobble together an ever bigger brain, just reanalyzing, analyzing, reanalyzing the same kind of information, just to squeeze a bit more out of it in terms of explicit knowledge. Right, so obvious question is, how could that have happened? Well, most of it happened very gradually. That's clear. If we look at, this is not time, by the way, although it's an approximation to time. It's just ranked. This is just a organ scale. We're looking at different species of monkeys, old world monkeys, new world monkeys, and then eight crosses here, and different species of hominids. Forensis through to erectus here. And you can see that you can rank, they're just simply ranked according to, well, something called encapsulation quotient. This is not size of the brain alone, it's size in relation to total body size. The argument being, with lots of evidence for this, that big bodies just demand bigger brains to analyse all the extra sensory information, control the extra muscles. So you'd expect brains to get bigger without any change in real competence with body mass. And that's been kind of factored out here. So this is sort of a genuine difference in brain size. So we've got over and above variations with body mass. And these are just rapid terms of that. And you see that the monkeys tend to be lower down here, the apes tend to be up here, and then the hominins are coming here. And then there's this. Just enormously different from that sequence. Double the equalization quotient of any other primate, and that, of course, is sapiens. In fact, recent work, very beautiful work by a Brazilian research woman. She developed a very simple technique, simply for fragmenting brains without losing the nuclei. It's just completely mushing up the brain, but every nucleus was intact. And then labeling the nuclei with the fluorescent tags according to their genetic expressions. So you label neurons as opposed to the endothelial cells, the blood vessels, and the and so on. And then run through a very rapid cell sorter and counting them. Just counting them. And what she found is that the human sapiens brain contains twice as many neurons as any other brain, including whale brains and dolphin brains. Well, certainly whale brains can be significantly bigger than brains, but their cells are larger and they're more sparsely scattered. So the human brain just has twice as many neurons as its nearest rival. It is really unusual. Just hold that in your mind. It is very distinct. It appears to be quite different from this population. All right. Now, I just want to address the question of how complexity might have evolved, and do this with some trepidation, which is the audience. The best way perhaps of sidestepping Richard is to quote the origin of all ideas. And Darwin was brilliant. This is a brilliant section of the origin because he anticipated the arguments against the ideas of natural selection. And nevertheless, those arguments, of course, were and continue to be produced. We focus on the eye. And this is a brilliant set of straw map. How could tiny variations, each individual being inherited, have contributed to the formation of a very complex organ, all the bits of which work together perfectly? It just looks designed. We know that different parts of the eye come from different bits of the embryo. And they somehow coalesce together, all mature perfectly, to produce this beautiful design. Neurals, mechanical, blood vessel, optical structure, which is the human eye. He sets it up, and then immediately, beautifully demolishes the process. And here's, of course, the classic argument. What matters is that you should be able to detect gradations within extant or fossil samples, as long as every type of preceding eye is useful in some ways to the animal hazard, even if it's those crude and poorly designed